All right, everyone, I think we can probably get started here. Thanks for joining us this afternoon for our webinar on Ignite, which is innovation grants to nurture initial translational efforts. We're going to kind of walk through the changes to the program and the NOFOs that we have and some general tips and tricks at the end, and then we'll take some questions. If you want to go to the next slide, Shruti. So I'm Becky Roof. I'm a program director for this program. We have here with us today Dr. Chardell Spriggs and Chris Conrad, who are both program officers who also hold some of these Ignite grants. Ms. Shruti Thomas and Dr. Cynthia Gunder are health program specialists working with us, and Ms. Ashley Gibbons is our operations coordinator. As I said, we're going to present the program and then answer questions. If your question is very specific to your particular project, please send us an Ames page and we'll answer that by setting up a call later. And that way we can spend time today on things that would be a general interest to everybody. We will have these slides and the recording posted on our website, so you can access that later if you miss something or if you want to share it with colleagues. Next slide. So the goal for Ignite is really to help people get ready for the next stage. This is early stage therapy development. We want to prepare you for programs such as the Blueprint Neurotherapeutics program, such as the Therapeutics Development Program. We've had people go on to urgent, private investments, small businesses, not-for-profit funding. And by the way, the Pain Therapy Development Program has a webinar exactly 3 o'clock right after this. So if you're interested in learning more, that's available. A lot of these programs still have webinars that you can check out if you want to learn more. Next slide, Shruti. All right, so we have in Ignite 3 funding opportunity announcements. The first is PAR 25255 for neurotherapeutic agent characterization and in vivo efficacy studies. We have one for assay development and therapeutic agent identification, and we have one for model development for the purposes of neurotherapeutic development. We're going to walk through each of these, but up front, I'll tell you that all 3 have some commonalities. They all have the same budget, up to $499,000 in any given year, with no more than $750 for the entire project, and that's in direct costs. We have 2 due dates listed here, but these are open for full 3 years, so there'll be plenty more to come. You can check the NOFO for those dates. I pretty much every time get questions about, hey, I'm a reviewer, can I submit late? And so here's the policy. You'll also notice that the A1 and the A0 have the same due date. If you've submitted an A0 to the prior issue of Ignite and you would like to submit an A1 to one of these new ones, you can do that because they are reissues, provided all the other pre-submission policies are followed. All right, so next slide. The R61, R33 mechanism is something that's new for a lot of people, so I like to take a minute to just describe what it is and how we use it. You will write your application with an R61, a milestone, and an R33. But if you get awarded, you will get an award for just the R61 up front. You'll do that work, you'll show us the data, and if milestones are met, then we award the R33. Why do we do this? We do this so that you can propose and we can fund dependent aims. We expect the R61 is kind of like set up, show feasibility, get your assays together, for example. And then on the R33, that's where you'll do the big experiment that gets you ready for the next step. Those dependent aims are de-risked by the milestone that's in the middle. So review is fine with those dependent aims. NIH is excited to fund dependent aims because we have those milestones in the middle, and now there's no risk, right, for us. But this whole thing only works because those milestones, right? That's the keystone to this whole thing. And so it's really important that milestones be really clear, that they be really quantitative, that they're justified, that they show that the R61 was successful and the R33 is worth doing. And by the way, review is really, really good at dinging milestones that are weak or poor or fluffy or low bar. Review historically for this program has been really, really good at calling that out. So be prepared, come in with really good, strong milestones, and hopefully you'll do well. Now, next slide, please. I'm just going to take a minute to kind of illustrate some of this because this is something that's new to a lot of people. And like I said, this is something that review's really good at noticing and picking on. So I'm just going to show you, illustrate just a couple examples just to get you the flavor. We do have more information on our website. We have more examples there. Please take a look. But a weak milestone might be, we're going to scale up our compound that we're going to need later. A stronger milestone is, we're going to specify how much we need, what kind of purity. A weaker milestone would be, we're going to do PK studies, you're going to pick the best one. Stronger milestone, you specify, what do you need? What kind of half-life do you need? What kind of exposure do you need? And by the way, that exposure is hopefully connected to something, your potency or something. You have some reason to believe that that would be meaningful, right? And so it's also justified. A weak milestone is we're going to fully optimize and validate our assay. Stronger is, hey, this is the Z prime we need. This is the DMSO tolerance we need. And, here's why we believe that's useful. And then, lastly, we're going to treat our animals with our compound and see decreases in seizures. Well, a stronger milestone is we're going to see decreased seizure frequency by X. We're going to see decrease in duration by Y. You can see where that's quantitative. And by the way, there's a lot of times people will put in, hey, we need to see a statistically significant change. That's quantitative, but a stronger milestone is we need to see a change of X because we think that reflects something that would be clinically meaningful, right? You can see where that's stronger. And like I said, review is over the years gotten really good at noticing these things and pointing these things out. So you've been warned. Hopefully you can now come in with strong milestones that do well in review. All right, for the next slide. All right, at this point, I'm going to turn it over to Chardell, who's going to talk about the first NOFO.
Becky. So the Neurotherapeutic Agent Characterization and in vivo Efficacy Studies funding opportunity supports research to determine if early-stage neurotherapeutics have sufficient biological activity to warrant further development. Therapeutic agents include both small molecules and biologics. We use a combination of parameters, including target engagement, PK, and of course, in vivo efficacy studies to determine the feasibility of the candidate therapeutic agents. So necessary entry criteria that you should be sure to illustrate in your application include novelty, such as new knowledge around targets, mechanisms, pathways, or therapeutic indications. Projects should aim to develop or repurpose therapeutics that are significant improvements over what's currently available. There must be a strong biological rationale for the intended approach, along with preliminary data or literature-based evidence supporting the feasibility and importance of your proposed approach. Preliminary data should be rigorous and from well-designed experiments. So there are links within the NOFOs that take you to resources that outline the most critical elements to meet that criteria of a well-defined experiment. Projects should include a description of the unmet need for the therapeutic agents and a clear justification for how the findings from these studies are relevant to treatments for disorders that are within the NINDS mission. Explain how the candidate therapeutic will be optimized and advanced toward later stages of discovery once the R33 phase is complete, and describe which patients would ultimately benefit from a successful therapeutic when delivered. Next slide, please. So some examples of activities for the R61 phase include those listed here. So while not an exhaustive list, the next two slides taken together will provide guidance for where to place your studies within the application. And we really encourage you to look at the funding opportunity for more details. Activities usually found in the first phase are preparation of the therapeutic agents to support your proposed activities, characterization of your therapeutic agent, This could, for example, include confirmation of purity, stability, and PK-ADME properties. Studies to develop dosage forms to support proposed PD measures, or in vivo studies, pharmacokinetic studies to determine direct or indirect therapeutic agent levels, studies to confirm that therapeutic agents reach and engage the target site, studies to confirm selectivity of the therapeutic agents. So limited studies to identify potential pharmacodynamic biomarkers specific for the purposes of developing the therapeutic agents to be tested in the application are allowed. Studies to inform design, refinement, and validation of the PD measure and/or in vivo efficacy models and testing procedures. So SAR to optimize the candidate therapeutic agent is no longer allowed, and we'll address that in later slides. At the end of the R61 phase, there are criteria that should be met to transition to the R33 phase. So please keep this in mind when structuring your application and specifically your milestones. If everything goes well, you structured your milestones around these activities and your milestones are met. What that will mean is that at the end of the R61, we should see successful completion of all necessary preparation and characterization of your proposed therapeutic agent, all PK studies, design, refinement, and validation of your PD markers. And you should also have a further refined and validated in vivo study design, such as an animal model that meets the NINDS rigor guidelines that will allow demonstration of dose and exposure responses. So once you're in that R33 phase, some activities that are usually conducted include, but are not limited to, pharmacodynamic and/or in vivo efficacy studies with your chemically and biologically characterized therapeutic agent, dose response activity with the intended route of administration that will be relevant to your future clinical studies, studies correlating your PK and PD measures, preliminary studies to assess very early safety. This does not include IND-enabling tox studies, and validation and replication of studies to confirm your observed results. There are many activities for drug development that are either not in scope of the IGNITE program or fall within another IGNITE funding opportunity. So my last slide with you, I will review some of these non-responsive activities. So please pay attention, as applications that include these elements will be considered non-responsive and will not be reviewed. And of course, see the funding opportunity for the complete list. This can be a really confusing point for applicants when they are deciding which funding opportunity to submit their research. And again, reach out to us. We'll be happy to meet with you and decide. So some later stage activities that are not covered are the medicinal chemistry optimization or structure activity relationship studies for small molecules or optimization of biologics, any IND enabling studies, as well as manufacture of therapeutic agents for IND enabling studies or clinical use. These fall under more advanced programs, such as the Blueprint Neurotherapeutic Networks program. Basic science activities are also not covered. Remember, we're a translational research program. Some of these studies include studies that are designed to establish proof of concept for a biological target or use of therapeutic agents to identify targets that are relevant to a disease. Studies of disease mechanism, and development of assays or probes to support basic understanding of disease or other basic research. And we would encourage you to visit the NIH R01 parent announcement for these activities. Other things that are not covered, applications with a significant focus, such as an entire aim on developing pharmacodynamic biomarkers or other biomarkers. Please consider the NINDS biomarker program. Development of devices and device-drug combinations. We would redirect you to the NINDS Translational Devices program. And projects that are entirely in silico are also not appropriate. So there are some activities that are covered in IGNITE, but come under companion funding opportunities, and you'll hear more about those in upcoming slides. Those include extensive development and validation of animal or ex vivo models, and screening and identification of novel therapeutic agents. And lastly, human subjects research, which was previously non-responsive, is now allowed as long as it's not a clinical trial. And so I'll say thanks for your time and turn it over to Chris to discuss the next funding opportunity.
Sure, Del. So I'll be introducing the announcement PAR 25059, and this is for assay development and therapeutic identification and characterization. So the goals of this funding announcement are to develop new translational assays, which are usually in vitro or ex vivo, and with ex vivo materials. And these efforts really should be useful for identifying and characterizing novel therapeutics. Again, this is a translational RFA, as with all of these. And so we want to make sure that these assays are not just, just identifying routine things. We want to be able to identify new therapeutic agents as well. And with that being said, the entry criteria for this is really having a very strong, novelty to your assay, and with that, you should have like a strong biological rationale or premise, to the disorder or disease that you're trying to model in that assay. And then, and also that assay can be including, target-based or a phenotypic assay as well. And the relevance for therapeutic development, you just want to make sure that you are, again, mimicking some processes that are in the disease or specific for identifying those therapeutic agents. So on next, please. So as with all these, there's a first phase, and listed here are just some of the activities that are that can occur in this type of NOFO or funding announcement. So this the R61 phase is just for the early development and validation of these assays to really think about, developing a testing funnel. So, how would you identify your therapeutics first with your primary assay and then maybe some subsequent assays as well, just to try to hone down into identifying a therapeutic. And so with some of those assays, they can be evaluating potency, specificity, selectivity, purity, identity, and also stability of therapeutic agents. And these can include like binding or other, functional assays. And the assays can also, as I said, can, if they're part of your screening funnel, rule out like screening artifacts, like if it's hitting another off target or something like that. And also the assays can be including biophysical characterization, like if you're looking for different changes in structure or like secondary, tertiary structure, or some kind of conformational change, or if you're going after something that happens in the disease state, like aggregation or oligomerization, that might be another example. But then you can also look at functional parts too, like with looking at cellular uptake or target engagement, transduction or infection. And in the case of a neurological disorder, and then downstream, with that in mind, just that downstream functional readout, having that worked out. And you want to be able to have all the reagents developed in this phase, like, of course, positive controls or any kind of negative controls. And then how would you show how you could identify, useful therapeutics as well. And that's really just part of the whole assay development. And you want to make sure that's part of that is, again, what this next line is just, what is the throughput, of your assay? Is it going to be, medium or high throughput or even high content, just depending on the library that you're going to screen. And as with a lot of things, if you're using human iPSC lines, you just want to make sure you review this NOFO just about any kind of special criteria you want to put into your grant, mainly in storage and trying to get funding for that. Next. And with all these, you want to have a successful transition from the R61 to the R33. And so what we would need to see in this particular transition is we'd like to see that assay validated and ready to go. And what that means usually is that you have all these criteria that have met, the assay's, stable, and then can run what kind of therapeutics through. And have you done any kind of different testing of, different vehicles like DMSO tolerance, or, what's the, how many wells can you do per plate, and all these other things that are just important features, like Z prime and confidence intervals and all these things that are part of all assay development. So you want to be able to have all these things worked out, just to make sure you don't have any artifacts and be able to interpret the data if you're going to launch this. And so with that being said, if you meet that with appropriate quantitative milestones, then you'll be doing activities, most likely in the case of an assay that is high throughput, you could do high throughput screening, or you could, be working with prepared libraries, that are very focused in a certain, chemical chemotype, or you can, or a certain area. And then you want to, be able to make this, screening platform or set of assays to really give you hits. And what that is just something that has a nice, robust signal that's above the noise, and that you can really call it a hit. And having that criteria too is also important. And you just want to be able to make sure that your therapeutic actually has the right properties as well. You want to be able to, it's not just fluorescent or something, that's part of your readout. You want to make sure you're able to screen that out as well. That can happen. But then you want to look at also, certain features, if you can do this, like metabolism, like these, ADME criteria, you can do some of these things like, looking at different absorption or metabolism, or different types of, matrices, whether it's plasma or, brain tissue or lysate or other tissue, you can look at this. And then assessment, of course, as I said, off-target effects is part of that screening funnel. And as what was in the previous RFA or RFA funding announcement, this announcement does not include the optimization of therapeutic agents. That is part of another funding program, the BPN program, or the small business program. Next. And just to help with, just a list here of just some areas that are potential examples of non-responsive areas. And that's, these are kind of a theme to all of these funding announcements that are part of the IGNITE program. And that's including, any kind of applications on later stage activities, like IND enabling studies, or, as I said before, since we're developing assays, we're not really optimizing drugs. So that is not included either. And then the inverse too, since you're developing assays, you're looking for something translational. So you're not really working in the basic science area. So any applications that are including just basic science work is not appropriate for this, and would be better suited, as mentioned prior, by an R01 mechanism or R21, or we have other mechanisms for that. And with that too, if you're planning to develop a therapeutic, look at one of our other programs, which was just mentioned about the efficacy models, and then also the one after this, which is in the model development. If it's doing any kind of those activities, you want to make sure that you're applying to the right one. This is primarily for assay development. And, as said before, we don't want to, double fund things. So anything that's really focused on biomarkers, there's programs for that in part of the NINDS, and then also our other, I guess, multi-IC programs like HEAL or other programs, they address this. And as I said, as was said before too, we don't develop devices. They have special programs for that, and special RFA funding announcements for those. And we do now allow funding subject research with materials coming from, human material. And we just have to check the RFA for the more details. So I'll just pass that off now to Shruti.
Hey, Shruti, can you unmute yourself?
Can you hear me?
Yes, thank you.
Okay. So the next funding opportunity in IGNITE is PAR 25060, or the development and validation of model systems. The goal of this is to improve translational relevance of animal models and ex vivo systems that can be utilized to facilitate the development of neurotherapeutics. So we're really hoping to develop tools that can be used in drug discovery and the development process. And this would be something that's just not only beneficial to your lab, but other investigators, and can benefit the whole field. There are two concepts that are important to this NOFO. One is internal validation, which is the consistency of the experimental design considering precision and reliability. And the second is external validation, which is the extent to which the results for which a study can be generalized from one setting to another, such as the preclinical to clinical setting. There are three different types of validities that we'd want to consider within external validation. Face validity is the similarity between the model or model system and clinical manifestation of the disease, such as symptoms or morphology. Construct validity is the similarity between model or the model system and the physiological basis of the disorder. So in terms of what is known about the disease mechanism. And the final validity is predictive validity, which is the similarity between the effect of a validated therapeutic intervention in the model or model system and in the clinical disease population. So this is referring specifically to a validated tool or therapeutic that has been shown to improve disease, to alter disease progression. Experimental therapeutics cannot be used to validate an experimental model. So we also acknowledge that there are plenty of neurological diseases that don't have therapeutics yet. And for these cases, it's not always possible or expected that you can evaluate predictive validity. Okay. So examples of activities for the R61 phase are initial development of the model or ex vivo system, including a matched control model if needed. One example is an isogenic control if you're using an iPSC model. And for this NOFO, you can use a model, you can develop a model completely from scratch, or you can use an existing model. And if it has poor validation, you can then improve the validity. Another example is any optimization related to feasibility, endpoint range, sensitivity, etc., and internal validation for endpoint use. By the end of the R61 phase, you really want to show that your model or model system is feasible for all the external validation studies in the R33 phase, and you want your milestones to address these points in a quantitative way. Examples of activities for the R33 phase include comparing phenotypes to human disease, comparing disease etiology in preclinical species to what is known about the human disease, and efficacy of clinically validated therapeutic agents if available in the new model system. Another example would be independent replication at another site. So if you had a collaborator at another university, this would be one example of that. Okay. Non-responsive studies for this NOFO include any application with activities aimed or covered for the efficacy and assay development NOFO. Applications that contain basic science activities such as studies of disease mechanism or identification of drug targets are also considered non-responsive. These studies would probably be more amenable to the R01 space. And like Chris and Chardell said, applications that focus on biomarkers and devices have their own programs housed in DTR. So we encourage you to check out those online. Right, and I will pass it on to Becky.
Hey, great, thank you. So I'm going to wrap it up by kind of giving some thoughts that apply to all three NOFOs. First is there's some changes with the reissue compared to the prior issue. For those of you who are familiar with what we supported in the past, a big one is that optimization is no longer supported for small molecules or biologics. This is, as has been mentioned already, this is supported by BPN, and if you are able to apply to one of the Blueprint Neurotherapeutics programs, you are better suited doing that because that will take you further. It will take you closer to the clinic. It will provide for you some resources that Ignite doesn't provide. It's really, if you're able to apply to that, I would encourage you to do that instead of Ignite. And I will point out, especially with the small molecule BPM, they've called out the need for some earlier stage stuff. So if you've heard in the past that it's hard to get in, yeah, not true anymore. They've revised the language and asked reviewers to consider that. So um, please consider that program if you're doing optimization. And we're defining optimization as the rational iterative design of new agents based on data obtained in a prior iteration. So think small molecule structure activity relationship studies. Um, it is okay to use predefined agents or a predefined library here for Ignite. The difference is it's predefined, right? Reviewers can evaluate what it is you're testing. We've heard already in human subjects research, we used to say things were not in scope or not responsive if they were human subject research unless they were in E4. We've just crossed that out. We now require a plan for enhancing diverse perspectives. There's a lot of resources the NIH has on this where you can learn more, and you can also read about it in the NOFOs. Milestones are now a separate attachment. They used to be embedded in the research strategy. Now it's separate, and hopefully that makes it easier for people to see that it's there or it's not, and hopefully if it's lacking, it's something you can address before you submit. We've I hope clarified the language about what is needed for a responsive complete application. That R61 and R33 are distinct activities that don't overlap in time. Inclusion of milestones is a responsiveness criteria. The scope we've heard a lot about in the PEDP. Now, non-responsive applications will be withdrawn, so please carefully read the NOFO and make sure you understand what's required of you. All right, next. All right, another big change is we now have the NIH simplified review criteria. This is brand new for us. Instead of having the 5 criteria, significance, innovation, approach, investigator, and environment, it's now been condensed into 3 factors. The first factor is importance of the research is, should it be done? Factor 2, rigor and feasibility is, how well will it be done? And these are both scored on a scale of 1 to 9. Using the same scale as what we've had before with review. And then the factor 3 is expertise and resources. And this is not going to get an individual score, but it's part of the larger score. And reviewers are going to be asked, is it acceptable or isn't? And if it's not, why not? Additional review criteria is sort of similar to how it was in the past. It can affect the overall score, but it doesn't get its own individual score. Additional review considerations. This is where the intellectual property is, and this is actually the same as what we've had before. It doesn't impact the score, but we ask reviewers to comment. A new feature is that some of the stuff that used to be part of peer review is now part of NIH internal review, things like the appropriateness of foreign work, data sharing. Even though this isn't peer reviewed, it's not going to be impacting your score at all. It's still really, really important and will be necessary for an award. All right, next slide. All right, so I'm going to close out with some just general tips. Please contact us in advance. We're here to help you. I know a lot of work goes into these, and if we can spend 30 minutes on the phone making sure that you're applying to the right NOFO and that you have the responsiveness, I think that's worthwhile. Include a rigorous study design and supporting data. NIH has been talking about rigor for a really long time, but I will say that this review panel has historically been really good at paying attention to this. And so I encourage you to also pay attention to this so that you can write a competitive application. Have a multidisciplinary team, and notice that it's a multidisciplinary review. So we haven't talked about review much yet. This is a NINDS panel. It is not reviewed at CSR with the R01s. It's got a special translationally focused review panel full of experts in disease, but also experts in pharmacology and drug development and gene therapy or whatever is relevant for your, reviewing your application. There'll be clinicians, there'll be academics, there'll be industry folks. It's very multidisciplinary, but it's also very translationally focused. They will be asking questions about where is this going? Do they know where they're going? Do they know what patient population would benefit? And are they working backwards from that appropriately? So know your audience and write an application that's going to appeal to this diverse panel. The best way to do that is to have a diverse group of experts on your team that help you write the application. You'll want to discuss intellectual property. This is also something we haven't mentioned yet, but it's uh, a requirement for these NOFOs if it's, except for the model development one. Have a therapy development plan. Also something that's important for the therapeutic NOFOs, not the model one. And this doesn't have to be super detailed, but you do need to convince reviewers that there's a path forward and that you're thinking about that. And that you know what obstacles you might encounter later and that you're preparing yourself for that. Another little feature about Ignite that people don't always realize is if you want to request a no-cost extension at the end of the R61, you can do that, but it requires prior approval. And this is just so that we can keep track of things and understand why we get these requests. The data management sharing policy, this is not super new anymore, but it might be new to some folks. The link here, you can check it out, make sure you understand it. That will apply here. If you are an SBIR eligible small business or an STTR eligible small business, I encourage you to use the small business program so that you can use the funds that NINDS sets aside for that program. We set aside money specifically for small businesses and only small businesses. So if that's something you can utilize, I encourage you to do so. Now, I get asked this all the time, so I'm just putting it up front here in the public space. And there's a link here you can look at historically what the fundable ranges have been for this program. Now, I can't stress enough, though, that this is historical. This does not predict the future. Things likely can change. Fiscal year 25 is looking particularly uncertain. So don't assume anything about the future, but this is historical funding ranges for this program, just to give you some reference. And again, look at that website for more details. All right, next slide. All right, that concludes the informational section. If you have questions, though, please, I see we're getting some already in the chat and in the Q&A. And then also reach out. My email's here if you want to talk about your AIMS page or you want to talk about an application. We're here to help and we're happy to talk to you. All right.
I can give you the first one.
Sure.
Mary Thomas asked, can you submit an SBAR in January and also an Ignite grant in February?
If they are overlapping in scope, you will run into the overlap policy. So it kind of comes down to timelines. If you have your summary statement for the SBIR, you might be able to, but that depends on the timing. But I'm also going to caution you here because it sounds like you're going to have a choice to make between applying to Ignite or resubmitting an SBIR if the first one doesn't look like it's going to get funded. A resubmission might be in your best interest if you feel like the issues are addressable and it's something that you want to have the opportunity to explain to reviewers, hey, this is how we fixed it. It sounds like you're being driven solely based on who has a sooner timeline, and there are other things to consider, like the focus of the NOFO or the ability to use set aside. So we can talk more about it if you want to, but don't make your decision just based on when the deadlines are. I guess is the bottom line there.
Okay, great. The next one is from Liquan Wang. For collaborating between 2 to 3 different PIs from different institutions, is the multi-PI mode allowed for this, for the PAR 25225?
Yeah, in fact, we encourage multidisciplinary teams, so absolutely go ahead and do an MPI if that's appropriate for your project.
And the second part of that is, will the award be made to the 2 institutes with separate base budget allocation or to the contact PI's institute only?
Yeah, it's handled the same way an MPI would be with an R01. So the grant would go to the contact or the submitting institution, and then they would have a sub award.
Okay. The next question is, Candace Strang, even though it was answered in the chat, is my understanding that Blueprint is a small molecule only, but not biologics? Am I in error here?
Oh, I'm glad somebody asked that. There are two Blueprint Neurotherapeutics programs. There's a small molecule one and there's a biologic one. They're structured really the same. But one is for biologic. So I'm glad you brought that up because yes, there are two.
Okay, great. And the other one is a human iPSC-derived organoid tissue chip model considered to be responsive to the model system NOFO or another NOFO?
Yeah, so as long as you're fitting other aspects of the NOFO, staying in scope, then absolutely that is a model you could develop in our model development NOFO.
Would that be in the assay development or the model development?
It depends what else you want to do with it. If you're spending 3 years to really build it and validate it, then I would encourage the model development assay or model development instead of the assay one. If you are mostly done and you're just going to tweak a few things and then use it as a high throughput screen, then you could, use it as an assay development therapeutic agent, um, NOFO. So it really depends on exactly what you want to do with it.
Okay. Another question is, can foreign collaborations be supported through this mechanism?
Yes.
Right. And I believe I've covered them all. I don't know if there's any further questions.
We can hang on a minute and see if anybody comes up with anything. Might take a minute to write them out. Oh, I see one we got, Chris. Who would be the person to contact for PIR 25? I'm thinking 060. Contact me first, I guess, and then we'll divide as necessary because it's based on the scientific space rather than the NOFO. Anything else? All right. If not, I guess we can conclude, but thank you everybody for your attention. Oh,
Thank
I
you
see
so
one more.
much.
Oh, sorry, I see one more before we close out. Um, can small businesses apply to Ignite? You are eligible, but as I said, if you can use the SBI program, that's encouraged just so that you can take advantage of the small business set aside. But small businesses are certainly eligible. Anybody else? All right. In that case, I think we'll close up. Thank you everybody for your attention. This is where you can reach out to me if you have further questions. Happy to talk about any project specific questions you might have. And look forward to hearing from you.